Situason, and I will report the mitosis and meiosis of group 3.
Name is Maria Arceli Moreno.
Mary Ann Sula.
Name is Rosane R. Padullo.
Miss Jessa Chudosho.
Janminelli Palaganas.
Let's start. First, what is mitosis? Mitosis is a process where a single cell divides in two identical daughter cells, or we call it cell divisions.
Cell division is a process of reproducing cells. It is necessary for organisms to undergo cell division because it's for growth, development, and repairing of tissues.
Mitosis is used to describe the duplication and distribution of chromosomes, the structures that carry genetic information. Mitosis consists of 4 basic phases: prophase, metaphase, anaphase, and telophase. In some textbooks, they have 5, breaking prophase into an early phase, which is called prophase, and a late phase called metaphase. These phases occur in strict sequential order in cytogenesis, or the process of dividing cell contents to make 2 new cells, which starts in anaphase or telophase.
The G2, the cell grows to double size and prepares for division.
When you look at this one, okay, before it begins, the mitosis, this cell is interphase has already copied the DNA. So the chromosomes and the nuclei each consist of 2 connected copies and called sister chromatids.
You can see the chromosomes very clearly at this point because they are still in their long, stringy dependence form.
In the prophase, the cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. The chromosome starts to condense, making them easier to pull apart later on.
So let's have the late prophase. In late prophase, sometimes called metaphase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes finish condensing, making them very compact. The nuclear envelope breaks down, releasing the chromosomes. The mitotic spindle grows more and some of the microtubules start to capture chromosomes.
After that, metaphase happens. So sa metaphase, the spindle has captured all the chromosomes and lined them up at the middle of the cell. So in metaphase, aligning of the chromosomes happens.
In metaphase, the chromosomes are lined up in metaphase plate.
So ayan siya, nakikita nyo nasa gitna sila lahat para mag-ready para mag-separate ito. At this age, the 2 kinetotures of each chromosome should be attached to chromosomes from opposite spindle poles. Before proceeding the anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate. And the kinetotures correctly attached to microtubes, this called spindle checkpoint, and help ensure that sister of chromatids will split eventually between the 2 daughter cells. When they separate, the next step, if the chromosomes is properly aligned, attached, the cell will have division until the problem is fixed.
In anaphase, the third phase of mitosis, the centromeres split and the sister chromatids are pulled apart to opposite poles of the cell.
The protein or glue that holds the sister chromosomes together is broken down, allowing them to separate. Each is now on its own chromosome. The chromosomes of each pair are pulled towards opposite ends of the cell. Microtubules not attached to chromosomes elongate and push apart, separating the poles and making the cell longer. All of these processes are driven by motor, proteins, molecular machines that can walk along microtubule tracks and carry a cargo. In mitosis, motor proteins carry chromosomes or other microtubules as they walk.
The next one is the telophase. In telophase, the cell is nearly done dividing. It then start and re-establish a normal structures are cytokinesis, division of the cell's contents, take place in.
So in telophase, the spindle fiber disappears, the chromosome starts to decondense, then the nuclear membrane starts to form again. Tapos, cytokinesis happens. So in cytokinesis, the cytoplasm splits into two, making a cleavage furrow. Then, after that, after the cytokinesis, we end up with two new cells, each with a complete set of chromosomes identical to those of the mother cells. Then, the mitosis ends.
Today, we will talk about one type of cell division, meiosis. What is meiosis? Meiosis is a cell division of sex cells, and it reproduces 4 daughter cells. Meiosis involves a sperm cell and egg cells.
Explains the function of meiosis. Meiosis is necessary for male sexual reproducing animal to ensure the same number of chromosomes is the offspring as the same as parents. The primary function of the meiosis is the production of the played in a number of chromosomes. The gametes from the ploid, 2N or the 2 sets of 23 chromosomes. The haploid is in or 1 set of 23 chromosomes. Problem during the meiosis, it can stop embryonic development and sometimes cause spontaneous miscarriage, genetically errors, and birth defects such as Down syndrome. Okay?
So just like in mitosis, before meiosis happens, the cells undergoes interphase, which is the G1 phase, S phase, and G2 phase.
So, we will go to prophase 1. As you can see on the diagram, synapses and crossing over occur in homologous chromosomes.
What happens in the prophase 1 is almost the same as in mitosis. Nuclear membranes or nuclear envelopes starts to disappear. Tapos, then the centrosome starts to produce microtubule spindle, and they start pushing apart a little from the spindles. Then the condensing of chromatins into chromosomes also happens in prophase 1. What the difference between the prophase of mitosis and prophase 1 of meiosis is that the chromosome or the homologous pairs of chromosome, meaning having the same gene code, undergoes crossing over. So what happens in the crossing over is that it's the sharing or genetic recombination of homologous pairs.
Okay? Meiosis 1, we call it metaphase 1. Homologous pairs of chromosomes line up to near the center of the cell. So, ayan, tulad yan, the kinetour microtubes from both spindle poles attach to their respective kinetours. The homologous chromosomes that contain the two different alleles from each genes are lined up to separate.
And next is anaphase 1. Homologous chromosomes separate and it's starting to divide and form a new cells.
Telophase 1, okay? The homologous chromosomes are pulled apart and moved to the opposite ends of the cell. The centrosomes do not dissolve, holding the sister chromosomes together. The cell elongates in the preparation of divisions down the center. That's all from the meiosis 1. Let's go to the meiosis 2, or we call it prophase. Okay? Chromosomes condense and the nuclei involved break down. The centrosomes are moved apart and spindle form between them, and the spindle microtubules begins to capture the chromosomes. So, ayan po siya. Ikita nyo naman po sa image. Okay?
And the next is metaphase 2. Dyads more to the center of the cells. A dyad consists of two sister chromatids.
In anaphase 2, the sister chromatids separates. So they are now called sister chromosomes and are pulled toward the centrioles. This separation marks the final division of the DNA. In telophase 2, the nuclear membranes form around each of the chromosomes, and the chromosomes decondense. Cytokinesis also happens, which is the splitting of the cytoplasm, forming the cleavage furrow of the cell. So, the final product of meiosis is 4 haploid cells in which each chromosome has just one chromatid.
Mitosis, when the cell splits apart. What is mitosis? Eventually, cells need to duplicate. There are two main methods of replication, mitosis and meiosis. This tutorial will talk about mitosis. The big idea to remember is that mitosis is the simple duplication of a cell and all its parts. It duplicates its DNA, and the two new cells, daughter cells, have the same pieces and genetic code. Two identical copies come from one original. Beyond the idea that two identical cells are created, there are certain steps in the process. Before a cell divides, it is in resting phase. The cell in resting phase is not actually resting, since it has to maintain its reservoir before it undergoes division. This phase is called interphase. It prepares the cell for division, i.e. protein synthesis, DNA duplication, and enzymes. After this phase comes mitosis, which is further divided into karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. Karyokinesis involves the 4 basic steps of mitosis that are prophase, metaphase, anaphase, telophase. Here we start with prophase. The chromosomes start coiling and become condensed. Nuclear envelope disintegrates. Gene transcription ceases during prophase and does not resume until late anaphase to early G1 phase. The nucleolus also disappears during early prophase. Centrosome gives rise to mitotic apparatus, i.e. generation of spindle tubules. Metaphase. Now all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the DNA, chromatin, has now condensed into chromosomes. 2 strands of chromosome are connected at the center of the centromere. The tubules connect to the centromere, not the DNA. Anaphase. Here we go. The separation begins. Half of the chromosomes are pulled to one side of the cell. Half go the other way. When the chromosomes get to the side of the cell, it's time to move on to telophase. Telophase. Now the division is finishing up. This is the time when the nuclear envelope begins to synthesize again along the separated chromosomes. Cytokinesis pinches the cell membrane in the middle. Cell membrane closes in and splits the cell into 2. Now we have 2 separate cells, each with half of the original DNA. Up till now, we have discussed what is mitosis and its different phases. Now it's important to know about significance of mitosis. Significance. It is an equational division through which identical daughter cells are produced, having the same amount and type of genetic constitution as that of the parent cell. It is responsible for growth and development of multicellular organisms from a single-celled zygote. The number of chromosomes remains the same in all cells produced by this division. Thus, the daughter cells retain the same character as those of the parent cell. It helps the cell in maintaining proper size. Mitosis helps in restoring wear and tear in body tissues, replacement of damaged or lost part, healing of wounds, and regeneration of detached parts, as in the tail of lizards. It is a method of multiplication in unicellular organisms. If mitosis remains unchecked, it may result in uncontrolled growth of cells, leading to cancer or tumour. The next important phase of cell cycle is meiosis. Have you ever wondered why you are different from your siblings? The answer lies within the amazing process of meiosis. What is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells, sperm in males, eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes than the parent cell. They are haploid. Meiosis takes place in germ or sex cells called gametes, eggs in females and sperm in males. Similar to mitosis, cells also pass through the interphase, G1, S, and G2 phase before they enter meiosis. Here are the distinct phases of meiosis. Interphase, meiosis 1, cytokinesis 1, meiosis 2, cytokinesis 2. First at interphase. In interphase, DNA is copied, resulting in 2 identical full sets of chromosomes to prepare for division. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. In next step at meiosis 1 takes place in following steps. Prophase 1. Prophase 1 is typically the longest phase of meiosis. During prophase 1, homologous chromosomes pair and exchange DNA, homologous recombination. The new combinations of DNA created during crossover are a significant source of genetic variation and result in new combinations of alleles, which may be beneficial. The paired and replicated chromosomes are called bivalents or tetrads, which have 2 chromosomes and 4 chromatids, with 1 chromosome coming from each parent. The process of pairing the homologous chromosomes is called synapsis. At this stage, non-sister chromatids may cross over at points called chiasmata, plural, singular chiasma. Prophase 1 has historically been divided into a series of substages, which are named according to the appearance of chromosomes. Leptotene. In this stage of prophase 1, individual chromosomes, each consisting of 2 sister chromatids, become individualized to form visible strands within the nucleus. Leptotene is of very short duration, and it's when the progressive condensation and coiling of chromosome fibers takes place. Zygotene. Chromosomes approximately line up with each other into homologous chromosome pairs through synaptonemal complex. The paired chromosomes are called bivalent or tetrad chromosomes. Pachytene. This is the stage when homologous recombination, including chromosomal crossover, crossing over, occurs. Non-sister chromatids of homologous chromosomes may exchange segments over regions of homology. At the sites where exchange happens, chiasmata form. Diplotene. The synaptonemal complex degrades and homologous chromosomes separate from one another a little. However, the homologous chromosomes of each bivalent remain tightly bound at chiasmata. The regions where crossing over occurred, the chiasmata remain on the chromosomes until they are at the transition to anaphase 1. Diakinesis. It closely resembles prometaphase of mitosis. The nucleoli disappear, the nuclear membrane disintegrates into vesicles, and the meiotic spindle begins to form. Synchronous process. During these stages, 2 centrosomes, containing a pair of centrioles in animal cells, migrate to the 2 poles of the cell. The microtubules invade the nuclear region after the nuclear envelope disintegrates, attaching to the chromosomes at the kinetochore. The kinetochore functions as a motor, pulling the chromosomes along the attached microtubule. Microtubules that attach to the kinetochores are known as kinetochore microtubules. Other microtubules will interact with microtubules from the opposite centrosome. These are called non-kinetochore microtubules or polar microtubules. Metaphase 1. Homologous pairs move together along the metaphase plate. The paired homologous chromosomes align along an equatorial plane that bisects the spindle. Anaphase 1. Kinetochore microtubules shorten, pulling homologous chromosomes, which consist of a pair of sister chromatids, to opposite poles. Non-kinetochore microtubules lengthen, pushing the centrosome further apart. The cell elongates in preparation for division down the center. Telophase 1. The first meiotic division effectively ends when the chromosomes arrive at the poles. Each daughter cell now has half the number of chromosomes, but each chromosome consists of a pair of chromatids. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid set. The chromosomes uncoil back into chromatin. Cytokinesis, the pinching of the cell membrane in animal cells or the formation of the cell wall in plant cells occurs, completing the creation of two daughter cells. Sister chromatids remain attached during telophase 1. Cells may enter a period of rest known as interkinesis or interphase 2. No DNA replication occurs during this stage. Meiosis 2. Meiosis 2 is the second meiotic division and usually involves equational segregation or separation of sister chromatids. Mechanically, the process is similar to mitosis, though its genetic results are fundamentally different. The end result of meiosis 2 is production of 4 haploid cells, N chromosomes, 23 in humans. The 4 main steps of meiosis 2 are prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, we see the disappearance of the nucleoli and the nuclear envelope again, as well as the shortening and thickening of the chromatids. Centrosomes move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase 2, the centromeres contain 2 kinetochores that attach to spindle fibers from the centrosomes at opposite poles. This is followed by anaphase 2, in which the remaining centromeric cohesion is cleaved, allowing the sister chromatids to segregate. The sister chromatids by convention are now called sister chromosomes as they move toward opposing poles. The process ends with telophase 2, which is similar to telophase 1 and is marked by the disassembly of the spindle and decondensation and lengthening of the chromosomes. Nuclear envelopes reform and cleavage or cell plate formation eventually produces a total of 4 daughter cells, each with a haploid set of chromosomes. Meiosis is now complete and ends up with 4 new daughter cells.